Good evening. Good evening. Am I audible? If you're joining the session for the first time, this is Dr. Shanmugapriya. I'm professor and head of the Department of Biochemistry at Government Thoothukudi Medical College. And today's session, it's session 2 of the series, wherein I plan to discuss biochemistry entirely in 3 hours. So this is for FMG aspirants, and I have picked up a few questions which are previously asked questions which are frequently asked. So topics and concepts based on previously asked questions have been chosen for today's session. And before we start the session, let me tell you a few facts about Unacademy plans. The first fact is about the NEET PG 2022 All India mock test, which is scheduled on May 1st. Good evening, Shivani. It is scheduled on May 1st and it is at 9 a.m. Yeah, it is at 9 a.m. And if you're asking me why should we attend this test, because this test will be, the question paper has been set in such a way that it would cover all subjects and it will have a blueprint which is expected, similar to what is expected in NEET PG. So that will help you understand exactly where you stand. And apart from this, there is also an All India rank prediction that is available for this All India mock test. So try to attend this test. And to enroll yourself for this All India mock test, you might need a code. You can use my code Shanmugapriya. And currently, apart from the Plus subscription and Iconic subscription, Unacademy also offers Lite subscription. And Lite subscription will enable you to access all the tests that are happening on the platform. We have multiple subject-wise tests, system-wise tests, full-length mock test, and grand test. So Lite subscription will help you appear for all these tests. And now NEET PG previous year question bank has been updated with last 4-year papers of NEET PG, INA, CT and FMGE. It is not just previous year questions. Every question is provided with a detailed explanation. So this will help you in the last run. I know you're all in the last lap of preparation for all your exams. So this previous year question bank section will be of great use to you. And this can be accessed by Plus subscribers, Iconic subscribers and Lite subscribers. And this can be accessed both on your mobile Unacademy app as well as on your laptop. And if you're an FMG aspirant, there is an FMG High Yield Revision MCQ batch. This is about to start on April 27th. And the duration of this course is 1 month. And in this 1 month, all 19 subjects will be discussed by all top educators using previous year questions and high yield topics and concepts. And this is the current NEET PG subscription plan. So to get Plus subscription, the minimum that you can opt for is 2 months. That would cost you 7,650. Iconic subscription, minimum duration is 6 months. That would cost you 36,000. Lite subscription, minimum is 2 months. That would cost you 2,160. And all these costs are inclusive of 10% discount. And to avail the 10% discount, you can use any educator's code. My code is Shanmugapriya. So that's all about Unacademy plans. Let's now start discussing these MCQs. I want all of you to be active during the discussion. Keep the live chat alive. So the first question is, which of the following proteins does not exhibit quaternary structure? What do you think is the right answer? Which of the following proteins does not exhibit quaternary structure? Hemoglobin, myoglobin, insulin and collagen. A quick clue that is given to you is only those proteins which have more than one polypeptide chain can exhibit quaternary structure. Only oligomeric proteins with multiple polypeptide chains can exhibit quaternary structure. In that case, do you think hemoglobin can exhibit quaternary structure? Yes, because hemoglobin has got 4 globin chains. Myoglobin has got only 1 globin chain. Very good, Yashas. Very good, Rids. So myoglobin with only 1 polypeptide chain cannot exhibit quaternary structure, whereas hemoglobin can. And insulin, don't you all know that insulin has got 2 polypeptide chains A and B connected by a C peptide. So insulin can exhibit quaternary structure. Collagen is a triple helix. It is made up of 3 polypeptide chains, right? It is a triple helix made up of 3 polypeptide chains. So which protein cannot exhibit quaternary structure? It is myoglobin because it has got only one polypeptide chain. Okay. Now the next question is, blood glucose at 12 hours post meal is maintained by. Can any one of you try to answer this? Blood glucose at 12 hours post meal is maintained by dietary glucose, muscle glycogenolysis, liver glycogenolysis, and gluconeogenesis. At 12 hours, it's predominantly maintained by liver glycogenolysis. Okay, so let's try to understand what are the ways by which we try to maintain plasma glucose. Plasma glucose can be maintained by three ways. The first one is dietary glucose, obviously, right? The first one is dietary glucose, and then it is liver glycogenolysis. The third one is liver gluconeogenesis. There are only three ways by which you can maintain plasma glucose. One is dietary glucose, second is liver glycogenolysis, the third one is liver gluconeogenesis. So initially, soon after you have your meal, dietary glucose will maintain plasma glucose, after which when the dietary glucose contribution maintaining plasma glucose decreases, liver glycogenolysis comes for the rescue. So this liver glycogenolysis begins 2.5 hours after the previous meal. When does it begin? It begins 2.5 hours after the previous meal. Suppose you have your lunch by 1 o'clock till 3 to 3:30. From 1 till 3 to 3:30, your dietary glucose will maintain plasma glucose. After which it has to be maintained only by liver glycogenolysis. And do you think this liver glycogenolysis can go on forever? No, because at one point liver glycogen will get used up. So liver glycogenolysis can maintain plasma glucose only till 12 to 18 hours of starvation. All of you please remember all these numbers. It is initiated 2 to 2.5 hours after the previous meal and can maintain plasma glucose only till 12 to 18 hours of starvation. So for the first 12 hours, what is the major source of plasma glucose? It is liver glycogenolysis. After which it is gluconeogenesis. And you will not wait for the last molecule of glycogen to get used up and then you will not switch over to gluconeogenesis. Gluconeogenesis begins well ahead. So gluconeogenesis begins 6 hours after the previous meal. Though it begins 6 hours after the previous meal, till 12 to 18 hours, the major source of plasma glucose would be only liver glycogenolysis. Is that clear? So the answer for that question is liver glycogenolysis. But I want you to understand few facts about glycogenolysis because this has been frequently asked in FMGE. So listen to this. Glycogenolysis, yes, fasting state. So glycogenolysis is done by which enzyme? Yesterday I told you in part 1 session, I told you glycogenolysis is done by glycogen phosphorylase. And what does the phosphorylase do? Phosphorylase adds inorganic phosphate and it cleaves glycogen. What is the phosphorylase? Phosphorylase mediates phosphorylysis in the sense it adds inorganic phosphate and it cleaves glycogen with n number of glucose residues to form glycogen n-1. What do I mean when I say glycogen with n becomes glycogen n-1? I mean to say the last glucose has been removed. And the last glucose that is removed will get added to the phosphate that is used for cleaving. So what is the product of glycogenolysis? Product of glycogenolysis is not free glucose, but it is glucose 1-phosphate. Are you all clear about this? Yeah, all of you please memorize this. Product of glycogenolysis is not free glucose, but it is glucose 1-phosphate. Now, what is the fate of this glucose 1-phosphate? Mutase acts on this glucose 1-phosphate and converts glucose 1-phosphate to glucose 6-phosphate. And until you form glucose 6-phosphate, steps involved in liver glycogenolysis and muscle glycogenolysis will be one and the same. From here, it's going to be different because liver being a gluconeogenic organ, because liver is a gluconeogenic organ, it is equipped with glucose 6-phosphatase. So only in liver, glucose 6-phosphatase can convert glucose 6-phosphate into glucose and that glucose reaches the circulation. So the carry home message here is only liver glycogenolysis can increase muscle glucose. Only liver glycogenolysis can increase plasma glucose. Muscle glycogenolysis cannot. And if you're asked why can't muscle glycogenolysis increase plasma glucose, your answer should be because muscle does not have glucose 6-phosphatase. Is that clear? Because muscle does not have glucose 6-phosphatase, muscle glycogenolysis can never increase plasma glucose. So blood glucose is maintained by all of the following except. For this question, what would be your answer? All of you answer this. Can blood glucose be maintained by dietary glucose? Yes. Can it be maintained by liver glycogenolysis? Yes. Can it be maintained by gluconeogenesis? Yes. But what cannot maintain plasma glucose? Muscle glycogenolysis cannot maintain plasma glucose. If you're asked why, what is your answer? Answer is glucose 6-phosphatase is absent in muscle. Clear? If it's clear, can I see a thumbs up, please? Okay, so it's always liver glycogenolysis, not muscle glycogenolysis, which can increase plasma glucose. Now, product of muscle glycogenolysis are all except. What is your answer? Product of muscle glycogenolysis are all except. Answer is glucose. Why not glucose? I already told you muscle glycogenolysis needs glucose 6-phosphatase for it to increase plasma glucose. Because muscle does not have glucose 6-phosphatase, it does not form glucose at all. Do you understand this? Suppose this is the muscle that you're looking at. So in muscle there is glycogen and muscle glycogenolysis gives rise to glucose 1-phosphate. Mutase converts it into glucose 6-phosphate. Very good. I'm very happy. Happiness, Paul, Sruzana, Rishnika, Javed, Sonali. Excellent. So mutase converts glucose 1-phosphate to glucose 6-phosphate. Because there is no glucose 6-phosphatase, what is the fate of glucose 6-phosphate that is formed in muscle? The fate of glucose 6-phosphate that is formed in muscle is that it gets into glycolysis. Do you all understand this? Glucose 6-phosphate gets into glycolysis. It gives rise to ATP. So product of muscle glycogenolysis will be glucose 1-phosphate, glucose 6-phosphate and ATP. It does not form glucose. Okay. Now this is an interesting question. Not just interesting, it's important. It is being repeatedly asked both in NEET PG as well as in FMGE. All the following are gluconeogenic enzymes except. Can anybody try answering this in the live chat, please? Pyruvate carboxylase, PepsiK, fructose 1,6-bisphosphatase and pyruvate dehydrogenase. Yeah, the answer is pyruvate dehydrogenase. Very good, happiness zone. Because have I told you yesterday, all of you please watch yesterday's YouTube session, live YouTube session. You can watch it as a recorded session. Because then I told you pyruvate dehydrogenase converts pyruvate into acetyl-CoA. And then this acetyl-CoA gets into citric acid cycle. So PDH or pyruvate dehydrogenase is not an enzyme of gluconeogenesis. And to remember the enzymes of gluconeogenesis, you have to know that gluconeogenesis is reversal of glycolysis. And to reverse glycolysis, you should know the irreversible steps of glycolysis. You should know a means of reversing them. So tell me, what are the irreversible steps of glycolysis? Yes, very good, Shruzana. It's a link reaction. Excellent. Aniruddha, very good. So tell me, what are the irreversible steps of glycolysis? The first step, hexokinase or glucokinase step. The second one is phosphofructokinase 1, the rate limiting enzyme of glycolysis. And the last irreversible step of glycolysis is pyruvate kinase step. All of you should memorize this. Yes, very good. So all of you please memorize this. The three irreversible steps of glycolysis are hexokinase or glucokinase, the first step, phosphofructokinase 1, the rate limiting enzyme, and pyruvate kinase, the last step. And to accomplish gluconeogenesis, which step will you reverse first? To accomplish gluconeogenesis, you have to reverse the pyruvate kinase step first. And usually pyruvate kinase converts phosphoenol pyruvate to pyruvate. Don't we all know that? Usually the last step pyruvate kinase converts phosphoenol pyruvate to pyruvate. So in gluconeogenesis, you should convert pyruvate into phosphoenol pyruvate. That is done by two enzymes. The first enzyme is pyruvate carboxylase. All of you memorize it. The first enzyme of gluconeogenesis is pyruvate carboxylase, which converts pyruvate into oxaloacetate. It converts pyruvate into oxaloacetate. And then to convert oxaloacetate into phosphoenol pyruvate, we use PEPCK. And what is PEPCK? As who answered it? Happiness Zone has answered it perfectly. PEPCK is nothing but phosphoenol pyruvate carboxy kinase. Okay, so that converts oxaloacetate to phosphoenol pyruvate. So tell me, what are the two steps? The first two steps of gluconeogenesis. First, it is pyruvate carboxylase, which converts pyruvate into oxaloacetate. PEPCK converts oxaloacetate to phosphoenol pyruvate. Now to reverse phosphofructokinase one step. To reverse phosphofructokinase one step, we use fructose 1,6-bisphosphatase. And to reverse hexokinase or glucokinase, you use glucose 6-phosphatase. So the blue color boxes, all these blue color boxes are the enzymes of gluconeogenesis. Clear? Pyruvate carboxylase, PEPCK, fructose 1,6-bisphosphatase and glucose 6-phosphatase. And additional information which I want you all to remember is pyruvate carboxylase is present in mitochondria. This has also been asked. Like pyruvate dehydrogenase complex, pyruvate carboxylase is also present in mitochondria. PEPCK is present in cytoplasm. Yeah, PEPCK and fructose 1,6-bisphosphatase are present in cytoplasm. Whereas the last enzyme, glucose 6-phosphatase, is present in endoplasmic reticulum. Glucose 6-phosphatase is present in endoplasmic reticulum. So gluconeogenesis involves multiple suborganelles. The first step happens in mitochondria. The remaining steps happen in cytoplasm. The last step catalyzed by glucose 6-phosphatase happens in endoplasmic reticulum. So if they ask you which is the microsomal enzyme marker, yeah, which is the microsomal enzyme marker, it means they're asking you which is the marker for endoplasmic reticulum. Your answer should be glucose 6-phosphatase. Is that clear? Which is the microsomal enzyme marker? It is glucose 6-phosphatase. So what is answer? All the following are gluconeogenic enzymes except, as all of you have answered it, is pyruvate dehydrogenase because it catalyzes the link reaction. A child presents with diarrhea, bloating, flatulence and frothy stools whenever he takes milk. The probable enzyme defect is. Before you start choosing these enzymes, can you tell me what is expected after taking milk if the person presents with GI disturbances? Shruzana thinks it's D. Javid thinks it's D. How many of you think it's D? So can we call it as lactose intolerance? All of you answer this. Shruzana, very good. A. Yes. Can we call it as lactose intolerance? Why am I calling it as lactose intolerance? Because it is caused by a defect of lactase. Yeah, because you see that clinically what you see is after the person takes milk, there is GI disturbances. So it's called as lactose intolerance. So what is lactose intolerance? It's caused by a defect of lactase. It is caused by a defect of lactase in the intestinal villi. So when intestinal villi does not have lactase, what will happen to the lactose that is present in the diet? All of you answer this. If intestinal villi does not have lactase, lactose in the diet cannot be digested. I hope you all know that lactose is a disaccharide. If you don't know it, memorize it. Lactose is a disaccharide made up of galactose and glucose. So when lactase is not present, lactose is not split into monosaccharides. If it is not split into monosaccharides, it cannot be absorbed. So lactose stays back in the intestinal lumen. And because lactose is osmotically active, it attracts water. Because lactose is osmotically active, it attracts water. So that causes what? That causes osmotic diarrhea. So the predominant feature of lactose intolerance would be osmotic diarrhea, because lactose is not getting digested, not getting absorbed, stays back in the lumen. Now this undigested lactose reaches colon. What will happen when lactose reaches colon? You know, colon is loaded with microorganisms, and these microorganisms utilize lactose to form hydrogen and methane. Intestinal or colonic microorganisms utilize the undigested lactose to form hydrogen and methane. Both hydrogen and methane are gases. So colon is loaded with gases, and that causes bloating. Yeah, that causes bloating. Very good, happiness zone. That causes bloating, that causes flatulence and frothy stools. Okay, so what is the reason for bloating? You don't have to write anything. Just listen to this, understand the concept. You can get it from the PDF. Everything is typed in the PDF. Now listen to it. So it presents with bloating, flatulence and frothy stools. The reason for bloating, flatulence and frothy stools is microorganisms utilize lactose to form hydrogen and methane, both of which are gases. Sometimes microorganisms utilize lactose to form acids. Yeah, sometimes they utilize lactose to form acids, and that is the reason for acidic pH of the stools. Yeah, and this acidic pH of the stool is the reason for, perianal, in the perianal regions, there will be rash and lactose intolerance. The reason for perianal rash is acidic pH of stool. So have you understood all about lactose intolerance? So all that you will have to remember about lactose intolerance is, following intake of lactose, there will be only GI disturbances. The child will not present with hypoglycemia. The child will not present with hypophosphatemia or lactic acidosis, because all those are features of metabolic defects. This is a digestion defect. So whenever there is a digestion defect, there will be only GI disturbances. What are the presentation? Osmotic diarrhea, bloating, flatulence, frothy stools, and acidic pH of stools. And because hydrogen and methane are getting generated, that is the basis. Yeah, because hydrogen and methane are getting generated, that is the basis of hydrogen breath test. So what is the investigation that you will perform to detect lactose intolerance? It is hydrogen breath test or methane breath test. All of you please memorize it. Hydrogen breath test or methane breath test can be done to detect lactose intolerance. Okay. So these are the features about which I have explained you. Okay. So now tell me the answer. I saw many of you to answer it as galactose 1-phosphate uridyl transferase. Galactose 1-phosphate uridyl transferase defect is called as classical galactosemia. What is it? That's called as classical galactosemia, and classical galactosemia will present only with metabolic defects like hypoglycemia, hypophosphatemia, lactic acidosis. Do you find any such features here? No. So this is not galactose 1-phosphate uridyl transferase defect. What is it? It is lactose intolerance caused by lactase deficiency in the intestinal villi. Okay. So now the next question. A child presents with hypoglycemia, hypophosphatemia, jaundice and hepatomegaly after transitioning from mother's milk to infant foods. The probable enzyme defect is, can it be lactase deficiency? All of you try to exclude choices. Just now I told you lactase deficiency is lactose intolerance, which presents only with GI disturbances. So we can exclude lactase deficiency straight away. Rishnika thinks it is D. Yeah, many of you think it's D. So can it be galactose 1-phosphate uridyl transferase defect? What is it called as? I told you it's called as classical galactosemia. And this is not classical galactosemia based on history, because history clearly states that only after transitioning from mother's milk to infant foods, all these have started appearing. Yeah, had it been a galactose metabolism disorder, it should have appeared even when the child was mother fed, because mother's milk has lactose, and lactose would have got digested to give glucose and galactose in the intestine, and galactose would have entered into the circulation even when the child was mother fed. So if it was a galactose metabolism disorder, it should have appeared even when the child was fed with mother's milk. because history clearly says only after transitioning to infant foods all these have started appearing. I want you to know that infant foods are rich in sucrose. Table sugar is sucrose. And infant foods, to make the infant like it, they load it with sucrose. Sucrose gets digested in the intestine to give glucose and fructose. After fructose has entered into the system, all these features have appeared. So it is hereditary fructose intolerance. Are you all clear about this? This is hereditary fructose intolerance caused by a defect of aldolase B. Okay, what is aldolase A defect? Can anybody answer about this? What about aldolase A defect? Happiness zone, now it eats fruits, honey or fructose based diet. Yes, it is predominantly infant food. Infant food, anything is rich in fructose. Okay. So aldolase A is an enzyme of glycolysis. If a glycolytic enzyme is defective, which cell gets affected the most? I want all of you to think and tell me, when glycolytic enzyme is defective, which cell gets affected the most? Yesterday in the session, I told you the cell which is totally dependent on glycolysis is RBCs. So any glycolytic enzyme defect, RBCs get affected and that will present as hemolytic anemia. Aldolase A defect or any glycolytic enzyme defect will present as what? Hemolytic anemia. Have they given you anything about hemolysis here? No. So this is not aldolase A defect. Aldolase B defect is the answer, which is hereditary fructose intolerance. Okay. So answer for this question is choice D. The micronutrient that is deficient following post-ileal resection is, because minerals are also often asked, minerals and vitamins. I thought I should give you more questions on that. So the micronutrient that is deficient following post-ileal resection is, can anybody answer this? Very good, Rishnika. Yes, it is B12. Yeah, it is B12. Sachin, it is not iron, because iron gets absorbed in the upper small intestine. Iron gets absorbed in the duodenum. Yeah, all these micronutrients, almost all essential very good. Happiness is right. So all essential micronutrients get absorbed in the upper small intestine. The only essential micronutrient that gets absorbed in the terminal ileum is B12. And not only B12, even bile salts. Yeah, B12 and bile salts get reabsorbed in the terminal part of ileum. So post-ileal resection, which vitamin will be deficient in a person? It is B12. Okay. And I want to tell you few facts about B12, because B12 seems to be a favorite topic of both FMGE and PG. So what are the facts that you need to know about B12? I want you to know that most of the sources of vitamin B12 would be non-vegetarian. It's predominantly non-vegetarian source that provides you B12, which means all these B12 in the source will be attached to proteins. Yeah, in non-vegetarian sources, B12 will be attached to proteins, which means, yeah. So which means when there is, when you want B12 to be absorbed, you should remove those proteins. And for that, you need proteolytic enzymes. What provides you proteolytic enzymes? It is pancreas. So one requirement for B12 absorption is pancreatic enzyme. The second requirement is intrinsic factor. So tell me, what are the two requirements for B12 absorption? One is pancreatic enzyme, which should remove off all the proteins. And the second one is intrinsic factor, because only after B12 is bound to intrinsic factor, it can be accepted by cobalamin receptors of terminal ileum. So if I ask you, what are the causes of vitamin B12 malabsorption? Can you tell me the answers? What are the causes of vitamin B12 malabsorption? It can be either pancreatic insufficiency. Yeah, it can be either pancreatic insufficiency or intrinsic factor deficiency. Intrinsic factor deficiency happens in type A gastritis, autoimmune gastritis with antral sparing. Okay, so that can be either pancreatic insufficiency or intrinsic factor deficiency. So if the question is, what is the test that you will do to detect vitamin B12 malabsorption? The test that is done to detect vitamin B12 malabsorption is Schilling's test. Do you all understand this? What is it? It is Schilling's test. Now, the other question that can be asked you about vitamin B12 is what are the coenzyme forms of B12? There are two coenzyme forms of B12. One is adenosyl B12. The other one is methyl B12. Yeah, one is adenosyl B12. The other one is methyl B12. So first about methyl B12, as the name indicates, it acts as a coenzyme for methyl transferase. Which methyl transferase is it? It is homocysteine methyl transferase. All of you, you have to know this. Yeah, methyl B12 acts as a coenzyme for methyl transferase. Which methyl transferase is it? It is homocysteine methyl transferase. As the name indicates, it converts homocysteine to methionine. It converts homocysteine to methionine. So now tell me what happens when there is B12 deficiency. When there is B12 deficiency, homocysteine methyl transferase will be inactive. So what accumulates in the blood? Homocysteine accumulates in the blood. So that causes homocystinuria. So one of the features of vitamin B12 deficiency is homocystinuria. Is that clear? And about adenosyl B12, that acts as a coenzyme for methylmalonyl-CoA mutase. It is triple M. Yeah, it acts as a coenzyme for methylmalonyl-CoA mutase, which converts methylmalonyl-CoA to succinyl-CoA. Yeah, it converts methylmalonyl-CoA to succinyl-CoA. So tell me what happens in B12 deficiency, which enzyme will be inactive? Methylmalonyl-CoA mutase will be inactive. So what accumulates in the blood? Methylmalonyl-CoA accumulates in the blood and that causes methylmalonic aciduria. Is that clear? What does it cause? It causes methylmalonic aciduria of B12 deficiency. And when methylmalonic acid accumulates, it goes and gets incorporated into myelin. Methylmalonic acid goes and gets incorporated into myelin and that causes abnormal myelination and demyelination. And that is why vitamin B12 deficiency will present with neurological manifestations. You all know that, right? B12 deficiency, they present with tingling sensation in the feet. Yeah, peripheral neuropathy. It starts as peripheral neuropathy and it ends as subacute combined degeneration. Do you all remember that? It ends as subacute combined degeneration. So what is the reason for all these neurological manifestations? It's only because methylmalonic acid accumulates. Okay. So if you suspect B12 deficiency, what are the manifestations that you expect? I want all of you to answer. B12 deficiency manifestations can be remembered using a mnemonic HMS. Have you all heard of hospital management system? Yeah, HMS is the mnemonic that you can have for, yes, loss of vibration, sense and proprioception, yes. So not only sensory pathway, even motor pathway gets affected and that's why it's called a subacute combined degeneration. So HMS is the mnemonic that you can remember to, that you can have to remember B12 deficiency manifestations. The first H stands for homocystinuria. Didn't I tell you? Yeah, the first H stands for homocystinuria because homocysteine methyl transferase is inactive. The second M stands for number 1 methylmalonic aciduria. Yeah, methylmalonic aciduria. Number 2 methylfolate trap. What is methylfolate trap, all of you? Whenever there is vitamin B12 deficiency, indirectly it causes folate deficiency. Yeah, whenever there is B12 deficiency, indirectly it causes folate deficiency, that is called as methylfolate trap. And when there is folate deficiency, there will be macrocytic anemia. So what are the three terms for M alone? For M alone, you have three manifestations. The first M stands for methylmalonic aciduria. Second, again, M stands for methylfolate trap. And then M stands for macrocytic anemia. Okay, and in macrocytic anemia, one of the features that you can see in peripheral smear is hypersegmented neutrophils. One of the features of macrocytic anemia that you can see in peripheral smear is hypersegmented neutrophils. Do you all remember this? This was asked in one of the FMGE papers or NEET PG, I'm not very clear. I think it was asked in FMGE. It was an image which showed you hypersegmented neutrophils and they asked you which deficiency causes it. Okay, so macrocytic anemia. And the last S stands for subacute combined degeneration. Last S stands for subacute combined degeneration. So have you memorized it? I hope you won't forget it anytime. B12 deficiency, what is a mnemonic? HMS. First H stands for homocystinuria. M stands for methylmalonic aciduria, methylfolate trap, and macrocytic anemia. And finally, S stands for subacute combined degeneration or neurological manifestations. Okay. So that's all about B12. The micronutrient that is deficient following post-ileal or following ileal resection is B12. Okay. So this is the question that I was referring to. A vegetarian patient presents with anemia and hypersegmented neutrophils. So there are so many clues that are provided to you here. Vegetarian means B12 deficiency is common with anemia, macrocytic anemia and hypersegmented neutrophils. So what is your answer? The most probable causes, yes, megaloblastic anemia by vitamin B12 deficiency. This is another question, previous year question. A person presents with diarrhea, dermatitis and dementia. What is the probable cause? Very good, all of you. I think everybody knows this. 3Ds of Pellagra. Yeah, what are the 3Ds of Pellagra? Diarrhea, dermatitis and dementia. Dermatitis particularly in the sun-exposed areas. Sun-exposed areas is around the collar. So that causes Castle's necklace. Okay. And it is caused by what? What is the cause of Pellagra, all of you? Niacin deficiency. So I'm going to now tell you causes of Pellagra. The first cause of Pellagra would be niacin deficiency. And apart from this, even in the yesterday's session, I was telling you I would not like to include the fourth D as death happiness zone because we as medicos are supposed to prevent it. Okay, diarrhea, dermatitis, dementia is what is acknowledged everywhere. Okay, so Pellagra is caused by niacin deficiency. And then do you all know that tryptophan can be converted to niacin? All of you, tryptophan can be converted to niacin. So another cause of Pellagra would be tryptophan deficiency. Another cause of Pellagra is tryptophan deficiency. Example, maize-based diet. Yeah, in maize there is tryptophan, but bioavailability of tryptophan from maize is very less. So a group of farmers, yeah, farmers, you know, they cultivate rice, they cultivate wheat, but they sell all their rice and wheat because those are costly crops. What they do is they rely on maize. So if a person is on maize completely, the tryptophan bioavailability is low, so that person can present with Pellagra-like manifestations. Okay, and even if there is tryptophan, yeah, even if there is tryptophan, what in the diet, what if there is tryptophan malabsorption? What if there is tryptophan malabsorption? That will also cause Pellagra. So tryptophan malabsorption is called as Hartnupp's disease. Yeah, it is Hartnupp's disease. Even if tryptophan is absorbed, it has entered into the system. And in the system, instead of tryptophan being used for niacin synthesis, what if tryptophan is used for something else? What is the other product that you get from tryptophan? It is serotonin. Do you all know that? Yeah, tryptophan can be used for both niacin synthesis and serotonin synthesis. So instead of it being used for niacin synthesis, if it's used for serotonin synthesis, that causes Pellagra. So carcinoid syndrome. Yeah, in carcinoid syndrome, major tryptophan that is available will be used for serotonin synthesis. So apart from Hartnupp's disease, it is carcinoid syndrome. And even if tryptophan is used for niacin synthesis, what if there is B6 deficiency? Yeah, B6 or pyridoxal phosphate is necessary as a coenzyme for kynureninase. B6 is necessary as a coenzyme for kynureninase. So whenever there is B6 deficiency, tryptophan cannot be converted to niacin. So that also causes Pellagra. So don't you think it's interesting to know that Pellagra is not only caused by niacin deficiency, it is also caused by B6 deficiency. The reason is B6 is necessary as a coenzyme for kynureninase, which converts tryptophan to niacin. Okay, and everything is perfect. There is tryptophan. It has entered into niacin synthetic pathway. There is also B6. But what if the diet is rich in leucine? If the diet is rich in leucine, leucine inhibits quinolinate phosphoribosyl transferase. There is an enzyme in niacin synthetic pathway which is quinolinate phosphoribosyl transferase that is inhibited by an amino acid leucine. So if the diet is rich in leucine, that inhibits niacin synthesis, that also causes pellagra, and that is called as leucine pellagra. And when will a person present with leucine pellagra? When the person is on sorghum. Yeah, sorghum based diet. So sorghum based diet will have will provide high availability of leucine, and that inhibits niacin synthesis, that causes pellagra. So that is leucine pellagra. So can you now tell me the causes of pellagra? I want all of you to tell me the causes of pellagra. Major cause which everybody knows is niacin deficiency. And then tryptophan deficiency, example maize based diet. And tryptophan is not getting absorbed. Thank you, happiness zone. So tryptophan is not getting absorbed. What will you call it as? You will call it as Hartnupp's disease. Okay. And tryptophan is absorbed. It is used for something else. What else can you use tryptophan for? Serotonin synthesis. So example, carcinoid syndrome. And tryptophan is used for niacin synthesis, but there is B6 deficiency. So tell me, B6 is necessary as a coenzyme for which enzyme? B6 is necessary as a coenzyme for kynureninase. Okay, so B6 deficiency. And finally, it is leucine pellagra. So when will there be leucine pellagra? Sorghum based diet, because leucine inhibits which enzyme? Quinolinate phosphoribosyl transferase. Okay. So person presents with diarrhea, dermatitis and dementia. What is the probable cause? It's very simple. It is pellagra. Menkes disease is caused by. What is answer? Menkes disease is caused by. A very simple question, right? Copper deficiency, copper toxicity, zinc deficiency, fluorine excess. I hope you all know that copper toxicity causes which disease? Wilson's disease. Right? Copper toxicity causes Wilson's disease and copper deficiency causes Menkes disease. So let me tell you few facts about Menkes disease. Menkes disease is caused by the defect of a protein. Which protein is it? ATP7A. Menkes disease caused by a defect of a protein which is ATP7A. Whereas Wilson's disease is caused by a defect of ATP7B. Do you all know that? Wilson's disease caused by a defect of ATP7B. If you're asking me how do I remember it? Between A and B, which alphabet comes first? A comes first. Yeah. And between M and W, which alphabet comes first? M comes first. So Menkes is caused by A defect. Wilson's is caused by B defect. Okay. That's how I remember it. And what is the role of ATP7A? It has got two roles. This transporter helps in copper absorption. One function of ATP7A is it helps in copper absorption along the intestine. Second role is the same transporter is also responsible for incorporating copper into enzymes. Yeah, the same protein is also responsible for incorporating copper into enzymes. Now tell me, what do you expect when ATP7A is defective? There will not be enough absorption of copper along the intestine. That causes copper deficiency. And there is no incorporation of copper into enzymes. What are the enzymes that need copper? One major enzyme that you should remember is lysyl oxidase. Lysyl oxidase needs copper. Not only lysyl oxidase, even tyrosinase. And then complex 4 of electron transport chain. So all these will need copper. So incorporation of copper into all these enzymes will need ATP7A. When that is defective, lysyl oxidase will be inactive. And do you all know that lysyl oxidase is involved in collagen cross linkages? Lysyl oxidase is involved in forming collagen cross linkages. So cross linkages are deficient. So connective tissue is weak. And this weak connective tissue is the reason for kinky hair. Number one. It is also responsible for repeated fractures and hypotonia. So what do you expect in Menkes disease? Yeah, in Menkes disease, you expect kinky hair, fractures and hypotonia, majorly because lysyl oxidase is defective, which is involved in collagen cross-linkage formation. So the carry home message here is, I want you to remember ATP7A and ATP7B, both are involved in copper metabolism. A defect causes which alphabet? M comes first. It causes Menkes disease. 7B defect causes Wilson's disease. Menkes is copper deficiency. Wilson's is copper toxicity. Okay. Yes, both vitamin C deficiency. Good, happiness. You're true. You're right. Cholesterol is synthesized from what? Cholesterol is synthesized from acetyl-CoA, bile acid, phenylalanine, heme. What do you think is the right answer? Cholesterol is synthesized from acetyl-CoA. Many of you have answered it as bile acid, not here. Generally, when I ask this question, many students answer it as bile acid, but I want you to remember that, yes, I can see one. Javed has answered it as B. Javed, from cholesterol, you get bile acid. Do you understand this? From cholesterol, you get bile acid. But the question is, cholesterol is synthesized from what? Cholesterol is synthesized from acetyl-CoA. Okay. So now let me tell you the various fates of acetyl-CoA. Acetyl-CoA is a central metabolite. It can have multiple fates. The major fate of acetyl-CoA is that it gets into citric acid cycle. When it gets into citric acid cycle, acetyl-CoA will come out as carbon dioxide. It gets completely burnt out as carbon dioxide. But if acetyl-CoA cannot get into citric acid cycle, do you know that oxaloacetate should be available for you to allow acetyl-CoA to react with it so that it can get into citric acid cycle. So for any reason, if oxaloacetate is not available, acetyl-CoA accumulates. And in that case, it condenses to form ketone bodies. Yeah, in that case, it condenses to form ketone bodies. So these two fates will happen when there is low energy. Do you all understand this? Whenever there is low energy in a person, the major fate of acetyl-CoA is that it gets into citric acid cycle, comes out as carbon dioxide, giving 10 ATPs. If it cannot get into citric acid cycle and if there is low energy, then acetyl-CoA condenses to form ketone bodies. Whereas when the energy status of the cell is high, when the energy status of the cell is high, you will want acetyl-CoA to get into anabolism. So we know acetyl-CoA is a building block of both fatty acid and cholesterol. Yeah, acetyl-CoA is a building block of both fatty acid and cholesterol. So when the energy status is high, acetyl-CoA is either used for fatty acid synthesis or for cholesterol synthesis. So now have you understood the various fates of acetyl-CoA? I hope it's clear. Yeah, when the energy status is low, it gets into citric acid cycle. Yes, happiness, you're right. It can get into citric acid cycle or it can form ketone bodies. When the energy status is high, it is used as a building block for fatty acid and cholesterol. Okay. What happens after a meal? This is also a previous year FMGE question. What happens after a meal? Will there be an increase in insulin, increase in glucagon, decrease in insulin, decrease in glucagon, or no changes in both? It's a very simple question, right? After a meal, what will happen to glucose availability in the blood? Increases. So this glucose gets into pancreatic beta cells. When glucose gets into pancreatic beta cells, what is released? Insulin is released. Right? Insulin is released and insulin will inhibit glucagon secretion. So following a meal, obviously there will be an increase in insulin and decrease in glucagon. Okay. Now this question I included here for this session because I thought you should know at least basics of how to interpret an ABG report. So I will not be getting into the details of it. I'm going to tell you just the outline that will help you in answering simple MCQs on ABG report. Okay. So a 60-year-old diabetic patient presented with repeated vomiting following a recent dine out. Before we start discussing about these values, can you tell me what is expected in vomiting? Which acid-base disorder is expected following vomiting? Vomiting, what is that you're losing? Following vomiting, you're losing gastric acid. Yeah, and not only gastric acid is getting lost, there will be dehydration. Vomiting causes dehydration. And what does this? Okay, I'm not asking you for answers. I'm happy that many of you are answering. But my question is, what do you expect in vomiting? Yeah. So following vomiting, there is dehydration, and dehydration causes secondary hyperaldosteronism. Aldosterone is released more. Secondary hyperaldosteronism. And this aldosterone causes sodium and water reabsorption. To counteract the dehydration, it causes an increase in sodium and water reabsorption. Reciprocally, it causes potassium and H plus secretion. So all of you please memorize it. Vomiting, alkalosis, happiness, you're partly right, you're partly wrong. Vomiting, yes, causes alkalosis, not because you're losing acid as HCl, but because there is dehydration, and this dehydration causes secondary hyperaldosteronism. And this aldosterone causes sodium and water reabsorption, but causes potassium and H plus secretion. When you secrete out H plus ion, what will it cause? It will cause metabolic alkalosis. Do you understand this? Yeah, that is why vomiting causes metabolic alkalosis. Whereas if the person is diabetic, a diabetic person is presenting with stress. Yeah, diabetes with stress can cause diabetic ketoacidosis. When there is diabetic ketoacidosis, what will you expect? You will expect metabolic acidosis. So based on clinical history, it can be either metabolic acidosis or metabolic alkalosis. Okay. Now let's try to solve this based on values. So there are few values which are given to you. My pleasure, happiness. So there are few values which are given to you. Before you look at these values, let me tell you normal values. Okay. So what is a normal pH? Can anybody answer this? Normal pH is 7.36 to 7.44. What is a normal PCO2? PCO2, you take off the 7. If you take off the 7, what do you have? 36 to 44. So normal PCO2 is 36 to 44 millimeters of mercury. Okay. And what is the normal bicarbonate? Normal bicarbonate is 24. On either side, you have 3. Yeah, 24 plus or minus 3. So what is it? It is 21 to 27 milliequivalents per liter. So have you memorized these three values? Normal pH is 7.36 to 7.44. Normal PCO2 is 36 to 44. Normal bicarbonate is 21 to 27 milliequivalents per liter. To interpret any ABG report, the first value that you should look at is pH. What's the first step? You look at pH. I said the normal pH is between 7.36 to 7.44. So what if the pH is less than 7.36? All of you answer this. What if the pH is less than 7.36? It means it is acidosis. If it is more than 7.44, it means it is alkalosis. But I want you all to know that normal pH of 7.4 does not exclude an acid-base disorder. Even if the person's blood pH is between 7.36 to 7.44, that does not exclude an acid-base disorder. Are you clear about it so far? Yeah, so this is step number 1, where you try to find out whether there is acidosis or alkalosis. After you find out whether there is acidosis or alkalosis, step number 2. Step number 2 is to find out whether it's a metabolic defect or a respiratory defect. Step number 2 is to find out whether it's a metabolic defect or a respiratory defect. And for this, you should look at bicarbonate and PCO2 values. You should look at bicarbonate and PCO2 values. I will give you an example. Suppose there is a patient with acidosis. This acidosis can be either because of low bicarbonate or because of high PCO2. Yes or no? This acidosis can be either because of low bicarbonate. Why do you think low bicarbonate causes acidosis? Because bicarbonate is a major plasma buffer. It has to buffer all metabolic acids. So for any reason, if there is low bicarbonate, metabolic acids are not getting buffered, and that causes metabolic acidosis. Do you all understand this? If there is acidotic pH and if it's accompanied by low bicarbonate, that is called as metabolic acidosis. Why do you think high PCO2? We were taught that pH below 7.2 patient will be dead, but in final year we saw patients and also in medicine textbook pH as low as 6.9 surviving. So as developments happen in medicine, yeah, viable values which represent viability will keep changing. Do you understand that happiness zone? Yeah, because development in medicine is tremendous, right? So previously we used to have few values for normal viability, but those normal values keep changing. So it is not like 7.2 the patient will be dead. Okay, so pH and PCO2 same side acidosis. I don't want you to have, okay, you can have any such mnemonics, but try to understand the concept. If you try to understand the concept, it'll be better. Yeah, because it'll be better for you to treat the patient also. It's not always just about answering an MCQ. Try to understand the concept, Paul Praveen Kumar. Listen to this. Why do you think high PCO2 causes acidosis? It is because, you know, carbon dioxide is a source of an acid, right? Whenever carbon dioxide accumulates, it reacts with water to form carbonic acid that dissociates to form H plus and bicarbonate. H plus is getting generated. So carbon dioxide is a source of an acid. So whenever there is high PCO2, there will be acidosis. And that is called as respiratory acidosis. Is that clear, Paul? Yeah, this is called as respiratory acidosis. This is about acidotic conditions. Now about alkalosis. Alkalosis can be either because of high bicarbonate or low PCO2. High bicarbonate causes alkalosis because bicarbonate neutralizes all metabolic acids. So that causes metabolic alkalosis. Low PCO2 causes alkalosis because carbon dioxide is a source of an acid. That is called as respiratory alkalosis. Are you clear about this? Yeah, are you clear about this flowchart? Yeah. So now can you look at these values and try to interpret it? So just start from pH. pH is 7.3. I said normal pH is 7.36 to 7.44. In this case, pH is low. So what is it? It is acidosis. Just because I found it as acidosis, what are the choices I'm going to exclude? I'm going to exclude B and C. Okay. Now this low pH, look at bicarbonate. Bicarbonate value normal is 21 to 27. Here bicarbonate value is low. So low pH is accompanied by low bicarbonate. Metabolic acids will not get neutralized. So what will you call it as? You will call it as metabolic acidosis. So why do you think this person is presented with metabolic acidosis? Because the person is a diabetic. The person is presented with a stress condition. So the person is presented with diabetic ketoacidosis. That is why there is metabolic acidosis. Is that clear? Yeah. So this is how simply you can try to solve all these questions. Now this is another question which was asked previously. All the following are required as coenzymes for PDH complex except B1, B3, B5 and B6. Can anybody try to answer this? PDH complex is frequently asked. Okay, so you have to know in and out about PDH complex. Okay, so the question is what are the following? All the following are required as coenzymes except. Very good, Aniruddha. The answer is B6. Yeah, B6 is not necessary as a coenzyme for PDH complex because PDH complex requires many vitamin B complex vitamins as coenzymes starting from B1 to B5. PDH complex requires B1 to B5 coenzymes except B4. B1 to B5 except B4. Why except B4? Because there is no B4 at all. We don't have B4 at all. So B1 to B5 and along with that you should also include lipoamide. B1 to B5 except B4. Along with that you should include lipoamide as well. Okay, so B6 or pyridoxal phosphate is not necessary. And for those of you who have forgotten what is B1, B2, B3 and everything, because many a times students forget it, let me quickly tell you that. What is B1? B1 is thiamine. What is B2? B2 is riboflavin. B2 is riboflavin. B3 is niacin. You can remember it as niacin has got many threes attached to it. B3 and 3Ds. Yeah, B3 and 3Ds. So B3 is niacin. B4, there is no vitamin B4. B5, 5 we call it as penta, right? 5 is generally called as penta. B5 is pantothenic acid. Okay, B5 is pantothenic acid. B6 is pyridoxal phosphate. B7 is biotin. There is no B8. So we don't have B4, we don't have B8. B7 is biotin. B9 is tetrahydrofolate. Okay, B9 is tetrahydrofolate. So what are the vitamins which are necessary as coenzymes for PDH complex? For PDH complex, it is B1 to B5 except B4 plus lipoamide. So what are the facts that can be asked about PDH complex? The first fact is, what does it do? You know, it converts pyruvate into, all of you answer this, pyruvate dehydrogenase complex converts pyruvate into acetyl-CoA. Second fact that can be asked you is, where is it present? It is present in mitochondria. PDH complex and pyruvate carboxylase are present in mitochondria. And then it uses 3 subunits. PDH complex uses 3 subunits. And what are the 3 subunits? The first subunit is named after the whole enzyme complex. What is it? It is pyruvate dehydrogenase. The second subunit is dihydrolipoyl transacetylase. The third subunit is dihydrolipoyl dehydrogenase. So all of you tell me what are the, so PDH complex, yes, happiness, you're right. It cannot happen in RBCs. You don't need it in RBCs also, because in RBCs it's only anaerobic glycolysis. Okay, so pyruvate dehydrogenase, dihydrolipoyl transacetylase and dihydrolipoyl dehydrogenase. So these are the 3 subunits of PDH complex. Now what are the coenzymes of these subunits? The first subunit uses B1 acids coenzyme. What is B1? Thiamine pyrophosphate. Pyruvate dehydrogenase uses thiamine pyrophosphate. Dihydrolipoyl transacetylase, the name tells you lipoyl. So what will it need? It will need lipoamide. Yeah, it needs lipoamide and CoA. So the second subunit needs lipoamide and CoA. That is why I told you, you need pantothenic acid. B5 is required for CoA formation, and that is necessary for the second subunit. And the third subunit, dihydrolipoyl dehydrogenase, uses FAD and NAD. It uses FAD and NAD as coenzymes. So tell me, what are the 5 so critical names difficult to remember? No, keep repeating it. You will be able to remember happiness. So tell me, what are the facts I told you about PDH complex? PDH complex converts pyruvate into acetyl-CoA, point number 1. Point number 2, it is present in mitochondria. Point number 3, it uses 3 subunits. What are the 3 subunits? E1 subunit is pyruvate dehydrogenase. E2 subunit is dihydrolipoyl transacetylase. And E3 is dihydrolipoyl dehydrogenase. And what are the coenzymes required? The first subunit uses B1 or thiamine pyrophosphate. The second subunit is dihydrolipoyl. So it needs lipoamide and CoA. The third subunit uses FAD and NAD. Clear? So that's all about PDH complex. So what is not required for PDH complex? It is B6. Okay. B6 is required as a coenzyme for all except, because the previous question was about all except B6. I want you to know what are the coenzyme roles of B6. It is required as a coenzyme for all except. Can you answer this? It is propionyl CoA carboxylase, because All carboxylases need biotin. Memorize it. All carboxylases, be it propionyl-CoA carboxylase or pyruvate carboxylase or whatever carboxylase it is, all these carboxylases need biotin. Okay. So B6 is required as a coenzyme for all except. So I want you to know what are the coenzyme roles of B6. The major coenzyme role of B6 is it is necessary as a coenzyme for glycogen phosphorylase. Yeah, B6 is necessary as a coenzyme for glycogen phosphorylase, an enzyme which is involved in glycogenolysis. And apart from this, B6 is necessary for all transaminases. You must have learnt about aspartate transaminase, alanine transaminase, right? So all those transaminases need B6. And not only transaminases, decarboxylases. Yeah, all decarboxylases also need B6. I will give you an example for a decarboxylation reaction. It is glutamate decarboxylase. So what does glutamate decarboxylase do? It converts glutamate to GABA. I'm going to tell you many clinical conditions based on this. So please listen to this and try to understand it. One example for decarboxylation reaction is catalyzed by glutamate decarboxylase, which converts glutamate to GABA. Don't you think it's interesting? It's interesting because glutamate is an excitatory neurotransmitter, whereas GABA is an inhibitory neurotransmitter. So you get an inhibitory neurotransmitter from excitatory neurotransmitter with the help of B6. And that is why seizures respond to B6 administration. Do you all know this? That is why seizures respond to B6 administration. Why? Whatever may be the cause of seizure, if you give a shot of B6, if you give a shot of B6 injection, any glutamate, any excitatory neurotransmitter that is present in the synapse will be converted to an inhibitory neurotransmitter, which is GABA. So that suppresses automaticity in the cortical neurons and that suppresses seizures. And this is why refractory seizure, for example, there is a neonate. Yeah, and neonate throws fits. The most common cause of neonatal seizure is hypoglycemia. The next common cause is hypocalcemia. Because you know hypoglycemia and hypocalcemia can cause seizure, you are starting calcium gluconate. Even after you start calcium gluconate, seizure activity is not suppressed. In that case, before you can find out the cause of seizure, you can try giving B6 injection. Seizure activity will be brought down. Is that clear? Yeah, so this is about glutamate decarboxylase needing B6. Another example for decarboxylation reaction is catalyzed by ALA synthase. How many of you have heard of ALA synthase? All of you, right? ALA synthase is an enzyme of heme synthesis. Yeah, it's an enzyme of heme synthesis. Okay, and this is why anemia responds to B6 administration. It's again interesting, right? Why do you think anemia responds to B6 administration? Not only to iron supplementation, anemia responds to B6 administration also, because B6 will stimulate ALA synthase, which is involved in heme synthesis. Okay, so this is about two examples for decarboxylation reactions. And then cystathionin beta synthase. Cystathionin beta synthase is dependent on B6. What is the role of cystathionin beta synthase? It converts homocysteine to cysteine. Yeah, if you want to convert homocysteine to cysteine, you need cystathionin beta synthase, which is dependent on pyridoxal phosphate. And that is why homocystinuria responds to B6 administration. So do you understand so many significances of administering B6? Yeah, that is why whenever you see a patient presenting with non-specific symptoms, first they start multivitamins. They start B complex vitamins in an attempt that something will get resolved. Okay. So B6 is homocystinuria. Yes. So whatever may be the cause of homocystinuria, it can be because of cystathionin beta synthase defect or because of other defect. When you start B6, Yeah, when you start B6 administration, homocysteine will get converted to cysteine and homocystinuria will respond to it. And the last enzyme that is dependent on B6, which you should know, is kynureninase, about which I already told you. Then I tell you to convert tryptophan to niacin. Yeah, to convert tryptophan to niacin, you need kynureninase, which is dependent on B6. And that is why not only niacin deficiency, even B6 deficiency can cause pellagra. And this is why pellagra responds to B6 administration. Clear? So can you tell me the coenzyme roles of B6? I want all of you to answer this. The most important coenzyme role of B6 is it is necessary for glycogen phosphorylase. And apart from glycogen phosphorylase, all transaminases and decarboxylases. And what are the examples for decarboxylases? I told you. Glutamate decarboxylase number 1. Number 2 is ALA synthase. And that is why seizures respond to B6 administration. Anemia responds to B6 administration. And then cystathionin beta synthase, which converts homocysteine to cysteine. That is dependent on B6. That is why homocystinuria responds to B6 administration. Kynureninase, which converts tryptophan to niacin, is dependent on B6. That is why pellagra responds to B6 administration. So can you all those who are here, can you now list out clinical conditions which respond to B6 administration? I want all of you to be active in the chat box. What are the clinical conditions that respond to B6 administration? The first one which I told you was seizures, neonatal seizure or any seizure for that matter. And then refractory anemia, seizure, anemia. And then homocystinuria, because cystathionin beta synthase will convert homocysteine to cysteine. And finally, pellagra, because kynureninase helps in the conversion of tryptophan to niacin. Okay, so that's all about B6. So now don't you think the answer is evident? I told you B6 is necessary for decarboxylation reactions. Yeah, B6 is necessary for decarboxylation reactions. So it is definitely not necessary for propionyl-CoA carboxylase. And I also told you carboxylases need what as a coenzyme? All carboxylases need biotin as coenzyme. Okay. Now another vitamin based question. Thiamine acts as a coenzyme for all the following except. Can you try answering this? Thiamine acts as a coenzyme for all the following except. It is transaldolase. Transaldolase does not need thiamine, whereas transketolase needs thiamine. Don't you already know that thiamine is necessary for PDH complex? You know that, right? Thiamine is necessary for PDH complex. And whenever you have PDH complex in any list, you can include alpha ketoglutarate dehydrogenase as well. So both PDH and alpha ketoglutarate dehydrogenase will need thiamine. Okay. And then transketolase needs thiamine. That is why whenever you suspect thiamine deficiency, what will you estimate? You estimate RBC transketolase activity. Yeah, whenever you suspect thiamine deficiency, what will you estimate? RBC transketolase activity. Very good, Aniruddha. So now I'm going to tell you about the coenzyme roles of thiamine. The first coenzyme role of thiamine is PDH complex or the link reaction. And not only PDH complex, even alpha ketoglutarate dehydrogenase complex. And then the third enzyme which I told you in yesterday's session, maple syrup urine disease, caused by the defect of branch chain keto acid dehydrogenase enzyme, right? That is also dependent on thiamine. And that is why I told you maple syrup urine disease responds to thiamine administration. Okay. And the last one is transketolase. So whenever you suspect thiamine deficiency, what will you estimate? You will estimate RBC transketolase activity. Okay. So these are the coenzyme roles of thiamine. So it is not transaldolase. Now, all the following are properties of genetic code except. Can anybody try answering this? All the following are properties of genetic code except degeneracy, ambiguous, non-punctuated, non-overlapping.